Good day everyone! In this video, we're going to talk about visual phototransduction. I have here the illustrations made by yours truly to explain how light is converted into neural signals and how the brain interprets these signals as color. Let's get started. Vision is one of the clearest examples of signal transduction in the human body. In vision, the stimulus is light, specifically photons, and the response is the perception of color, shape, and movement. This process involves multiple biological levels, from the eye as an organ, to the retina as tissue, to photoreceptor cells, and finally to molecular signaling pathways. At the organ level, vision begins in the eye. Light enters the eye through the cornea and passes through the lens, which focuses the light onto the retina at the back of the eye. The eye's main function is not to interpret images, but to capture light and deliver it accurately to the retina. At this level, the stimulus is physical energy in the form of photons. The retina is a specialized neural tissue composed of several layers of cells. These include photoreceptors, bipolar cells, and ganglion cells. The retina performs early processing of visual information before signals even reach the brain. The most important cells for detecting lights are the photoreceptors, which are divided into rods and cones. Rods are responsible for vision in low light, while cones are responsible for color vision and sharp detail. Color vision depends on three types of cone cells. S-cone respond best to short wavelengths of light, which we perceive as blue. M-cones respond to medium wavelengths, perceived as green, and L-cones respond to long wavelengths, perceived as red. Each cone type contains a different light-sensitive protein that allows it to respond selectively to certain wavelengths. Color perception depends on the combined activation of these cones rather than the activity of a single cone alone. Now let's dig deeper to the cell and molecular level. Again, photoreceptors are specialized neurons designed to respond to light. Each photoreceptor has an outer segment packed with light-sensitive proteins and an inner segment that generates electrical signals. Unlike most neurons, photoreceptors are active in the dark. Ion channels that are triggered by a chemical known as cyclic guanosine monophosphate or CGMP allow positively charged sodium ions to enter rod cells in the dark. This influx of positively charged ions causes cells to remain in a depolarized state, leading to the continuous release of the neurotransmitter glutamate. Inside the rod cell, there is a substance called rhodopsin, which is made up of a protein called opsin and a molecule called retinal, which is capable of absorbing light. When the retinal absorbs light, its configuration changes, an event that prompts opsin to activate a protein called transducin. Transducin stimulates a type of enzyme called phosphodiesterase, which starts to degrade CGMP. When CGMP levels decrease, the ion channels activated by CGMP start to shut. As a result, lower sodium levels enter the cell, causing it to become hyperpolarized from potassium ions that consistently exit the cell. Consequently, the release of glutamate reduces. Interestingly, this reduction in neurotransmitter release serves as an indication that a light stimulus is detected. The rod cell swiftly returns to its normal state once activated rhodopsin is inactivated, followed by the binding of a protein known as arrestin. Arrestin inhibits rhodopsin's capacity to activate transducin, preventing the cascade from proceeding. A complicated process subsequently returns retinal to its initial arrangement, preparing it to capture light again. The change in electrical activity alters the release of the neurotransmitter glutamate from the photoreceptor. Bipolar cells detect this change and relay the signal to the ganglion cells. The axons of ganglion cells then form the optic nerve, which carries the signal to the brain. Once it reaches the brain, the signal is processed in the thalamus and then sent to the visual cortex in the occipital lobe. In the brain, visual signals from different cone types are compared and integrated, allowing us to perceive color. Vision therefore is not completed in the eye but in the brain. So to summarize, vision is a signal transduction process where photons stimulate photoreceptor proteins, activate molecular cascades, alter cellular electrical activity, and ultimately produce color perception. From organ to tissue, to cell and to molecular signaling, Vision demonstrates how external stimuli are transformed into meaningful sensory experience. And that's all for visual phototransduction. I hope you've learned something and see you next time.